Make sure that's going. Fantastic. So that being said, as we move into kind of tonight's proceedings, as well as, talking about community, we'd first off just like to take this opportunity to acknowledge the traditional territories of the people of the Treaty 7 region in southern Alberta, which includes the Blackfoot Confederacy, as well as the Tsuut'ina First Nation and the Stoney Nakoda. The city of Calgary is also home to Métis Nation of Alberta Region 3. And we'd also like to note that the Alberta Lymphedema Association was founded on land adjacent to where the Bow River meets the Elbow River, and that the traditional Blackfoot name of this place is Mohkinstsis, which we now call the city of Calgary. So with that, we invite you all tonight and moving forward to really, think about what community means to each of us and recognizing our place in our history with the land and with each other as we move towards, peaceful reconciliation. So we look forward to now kind of moving into our proceedings for this evening. And I'd like to introduce our speaker tonight, Dr. John Conley. So Dr. John Conley is a professor and former head of the Department of Medicine at the University of Calgary and Alberta Health Services. He's medically trained as a specialist in infectious diseases and was a past president of the Canadian Infectious Disease Society, past chairman of the board for the Canadian Committee on Antibiotic Resistance, and a previous vice chair for the Canadian Expert Drug Advisory Committee. He is currently seconded at a half-time FTE to the World Health Organization Health Emergencies Program as a senior technical officer for COVID-19, the chair of the World Health Organization Infection Prevention and Control Research and Development Expert Group, co-director for the Snyder Institute of Chronic Diseases at the University of Calgary, and a member of the Canadian Expert Advisory Group on Antimicrobial Resistance, and a member of the Cochrane Collaboration Acute Respiratory Infections Working Group. He's published over 300 papers and has received multiple career honors in teaching, research, mentorship, innovation, and service, including the Ronald Christie Award for Outstanding Contributions to Academic Medicine in Canada, the Medal for Distinguished Service from the Alberta Medical Association for Outstanding Personal Contributions to the Medical Profession, as well as the Order of Canada. He continues as an active consultant in clinical infectious diseases with current interests which focus on antimicrobial resistance and stewardship, prevention of hospital-acquired infections, and novel innovations in healthcare. So with that, I'd like to welcome Dr. Conley, first off to share his slides, and once again reminding any members who may have their cameras still on, if you're able to turn off your camera for the time being, we welcome Dr. Conley to the floor.
Thank you very much for the very kind words. Just going to put my slides up. I have stepped down as the co-director of the Snyder Institute, so that's a past role. I think it must have been a previous bio that had been sent in, but this fall I had stepped away from that position after many years. I'm delighted to be able to speak to the organization tonight. I was asked to focus on COVID-19, and a topic that I've chosen is with respect to vaccines. And you can see the Department of Pediatrics up there, but that's actually the Department of Medicine. But I borrowed several of the slides from a colleague who is on the vaccine task force, who is with the Department of Pediatrics. But I'm delighted to be able to speak to you. I've tried to focus several of the slides related to lymphedema specific items. And Catherine has already done a territorial acknowledgement, so I think I'll move past this slide. I will state that I have no conflicts of interest with respect to this particular presentation. I do have one item that I was engaged in a study sponsored by Pfizer. They are the makers of one of the vaccines that I will be touching on, so I will point this out. It was a Staph aureus vaccine, and we were engaged in Alberta Health Services and the University of Calgary with administering the Staph aureus vaccine in a clinical trial for which I received no personal remuneration. All of the money went to the University of Calgary research offices to help hire the research personnel to conduct the clinical trial. That vaccine failed, unfortunately, but that's another story. It might be relevant to those of you with lymphedema who have recurrent staphylococcal or streptococcal infections, but perhaps for another time in the future. Just as an acknowledgement, my colleague, Dr. Jim Kellner, is a pediatrician and is engaged in the Canadian task force that is looking at the COVID vaccinations. He's in the Department of Pediatrics, and he kindly lent me several of his slides, so I wish to thank him for providing me with some of the slides. Here's some resources for you who are interested. The COVID Immunity Task Force, and they actually do a lot of the discussions about what's required in the vaccinations. Alberta Health Services has got a nice page, and they give you regular updates on who has received the vaccination. Then there's the COVID Tracker Canada, which is the federal government's vaccination tracker. And then faculty of Cummings School of Medicine website with a group with which I'm engaged, because I'm not only a member of Snyder, but also the O'Brien Institute. And this is a publicly available website, and they provide tracking of the new SARS-CoV cases, and they're going to be adding in a little addition with vaccinations and who has received them and what the daily count of additional vaccines is. So there's a rich source of information to be able to collect it. They also allow you to be able to compare multiple countries with respect to, and other provinces with respect to Alberta, where we're at, what is the slope of the curve, and it's really a wealth of information. I've been engaged with that group since the March of this year. One thing I wanted to say is that this is the first pandemic in which we've had instant social messaging, and this has been a problem because there's a torrent of social media and mainstream media, YouTube videos, Twitter, TikTok, many platforms in which information comes, and as soon as there's even a little bit of a tweak of information, it's suddenly blasted out on Dr. Google, and it's been very difficult for many in the lay public to be able to distinguish what's real and what's fantasy. So sometimes even with the papers that are published, the science may be correct, but there are jumps to premature or incorrect conclusions. And there have been more retractions in top quality journals since the spring of this year than in any other year in the past several decades. There was just actually recently a publication that was bemoaning the quality of some of the papers that were published with respect to COVID. And I don't have one of the slides, but there was one from New York City about the fact that there was a wind of air sweeping across the city, which led to all of the cases of COVID, which of course we know is not the case, but that was one of the reports. So you have to be very careful. And there's a lot on social media accounts about misinformation with respect to the vaccines, and I'll touch on that a little bit later. There are many letters, short reports. Many editors of journals want to be fast and first, and so you have to be very careful, and I suggest that you use some appraisal skills and also a good dose of common sense when you're reading some of the information. Just to remind you about new vaccines in normal times, vaccine development to phases of human testing takes about a decade. National regulatory review takes months to years, and that goes through the Health Canada approval, and then the Public Health Agency of Canada, and then finally the National Advisory Committee on Immunization. I was a member of that for a number of years and have stepped away. And then provincially, you have scheduling, safety standards, and that takes months to years. So there's no question that what we've seen happen with respect to the COVID vaccine program has been unbelievable. It's been an incredible journey over this past year. And my slide is not moving forward. There we go. So COVID vaccines in Canada, if we look at what's occurred now, you had vaccine development based on existing science, adaptive clinical trial designs, which Pfizer and Moderna did. You had in the United States, the rapid system warp speed, as they called it. You've got the COVAX alliance internationally, and multiple countries were moving at a breakneck pace to be able to develop the vaccines, engage in the clinical trials, and then the production. And even as I speak, there are ongoing trials in pregnancy and also in the setting of children with these new vaccines. So with respect to the purchase agreements and the rolling submissions, and then the preliminary National Advisory Committee guidance, it took just months. And then provincial scheduling has taken weeks to months, and suddenly we've got vaccine. And if you just think no one was looking at this back in February of this past year, some of the ideas were percolating, and I'll touch on that in a moment. But this has been an unbelievable journey that normally would take a decade at least, and perhaps a decade and a half, that we've seen the development of a vaccine. And if we look at the question that you'll see sometimes on social media, is it too rushed? Are they safe? If you look at the scientific expertise and some of the science globally that's been placed in the COVID epidemic, which has consumed much of my life in the last 11 months, since I had the first call from the WHO back in January of 2020, asking me to get on a call at about 2 a.m. in the morning about this new virus, and my world's been turned upside down ever since. But at the Shanghai Public Health Clinical Center, the genome was sequenced, working around the clock from clinical samples, and released publicly on January 11th. So that was done within 40 hours of very intense work between the 3rd and the 11th. The vaccine types for the messenger RNA vaccines, the concept was developed off an existing platform. So this was a vaccine that they say was 30 years in the making, but within 2 days they had mapped out how they were going to design this vaccine based on messenger RNA. And that's where the term Moderna, the M stands for messenger in this. And there was also frequent events that were occurring leading to the critical progress in the clinical trial delivery in multiple countries around the world. And then there was a public-private partnership with respect to the funding. And there are, you can see a couple of websites that I've listed at the bottom there if you wanted to track the vaccine development. And there's also the New York Times London or New York Times Vaccine Tracker. There's also one with the London School of Tropical Medicine and Hygiene. And then that's a scientific one. The New York Times is more of a lay scale type of tracker. But you can see what's happened is that you've got phase 1, phase 2, 3 trials that have gone on with respect to the testing, and now 2 vaccines approved for use with the messenger RNA vaccinations with Pfizer and Moderna, and now the AstraZeneca, although that is in the UK. And 1 vaccine was abandoned, which I'll touch on. As of December of 2020, the WHO and their landscape indicated that there were 52 vaccines in human trials yet, and 162 in preclinical evaluations. So there's still a lot of vaccine work going on. With respect to the pipeline, there are limited emergency approvals prior to completed phase 3 trials. There were 3 from China. You may have seen these, the Sinovac, Sinopharm, Wuhan, and the CanSino Bio, and then they were viral vector vaccines inactivated, and then 2 from Russia, the Sputnik 5 from the Gamaleya Institute, and then 2 a peptide-based vaccine as well. They were actually initiated in Russia prior to the phase 3 trials. Most of the phase 3 trials are on clinicaltrials.gov, so you can actually find them on the website if you look. And then the full approval after phase 3 trials, phase 1 being that they do the initial testing on volunteer subjects after the animal model studies, and then they do a phase 2 trial, which is to look to see the immune response with these vaccines and safety issues. And then phase 3 is where they do a comparative analysis, those that receive the vaccine, those that get the placebo or the normal non-vaccine containing components. It's like a sham, and then you compare the results between the 2. So we have the Sinopharm Chinese vaccine that's been approved after phase 3 trials, and then the Pfizer BioNTech and the Moderna vaccines, which are also approved. And they are in multiple countries around the world. I'm not going to go through them. What I've listed here is probably already out of date. Not all the vaccines have worked out. Australia abandoned their homegrown coronavirus vaccine. They had one that had HIV false positives, and GSK had a delay in their vaccine with their recombinant protein-based vaccine, and then also Sanofi as well. So not all of them are panning out, and that's important to recognize. We only hear about the ones that have made it to market and how can we get them in, but a lot of work going on behind the scenes. And December the 9th, a day in history in Canada, where Health Canada approved the Pfizer COVID vaccine, which is a messenger RNA vaccine, which I'll touch on in a moment. And just to spend a moment, this is a complicated slide, but there's the companies on your right, you can see, and then on the left, the types of vaccines. So it's important to note that the majority of these are non-live vaccines, and you can see the types with the messenger RNA. It's a non-virally active vaccine. You have the non-replicating viral vector vaccines, and these are unknown previously, so this is new technology. You can see Pfizer and Moderna are messenger RNA, AstraZeneca, Janssen, Johnson & Johnson, and the Sputnik 5 are the non-replicating viral vector. Then virus-like particles, this is not a COVID vaccine, but that's the human papilloma virus vaccine, the HPV. Protein subunits, which are used for whooping cough, for example, and hepatitis B. Inactivated whole virus, which is the polio virus. And then a replicating viral vector, which is the Ebola virus. And then there are a number of DNA viruses that are in development, and several in the COVID era as well. So you can see that there are a lot of different technologies being applied. And within the Canadian context, there are numerous vaccines still undergoing trials within Canada. There's a phase 1 trial completed and registered, not completed, but registered nearing completion, the Medicago. There are others pending. There was one up in University of Alberta, one at Vito in Saskatchewan. So there are a number of other sites across Canada. And then with the vaccine manufacturing capacity, the Montreal Centre, they're hoping will have capacity for a homegrown production of vaccines at up to 2 million doses a month. And Canada has also contributed somewhat to the COVAX initiative. This is a $2 billion fund set up with the World Health Organization to finance and procure vaccines for low to middle income countries. One area of controversy that you should all be aware about, if you from a social conscience perspective, is the high income countries have bought up all the world's vaccine, and there are countries in Africa and Asia that have no ability to purchase vaccines, and hopefully the high income countries with the COVAX initiative will be able to allow the vaccinations to be purchased by low to middle income countries, much like the HIV or AIDS drugs have been made available through special commitments. Canada, you'll see, has paid, and this is something we haven't been able to find out, how much money as the Canadian government spent on grabbing those 29 million doses, and another 20 million announced yesterday. And if you look at the bilateral agreements in Canada, they have been on a tear. You see Pfizer, Moderna, Medicago, AstraZeneca, J&J, Novavax, and Sanofi, all in the anywhere from 20 up to almost 80 million doses of vaccine that have been pre-purchased for Canada. And if you look at this was a figure that I was astounded about. It's a difficult one to find, but you can see here that Canada leads the world in terms of the number of doses purchased per citizen. So you can see that Canada has overtaken Australia and the United Kingdom, the United States, in terms of the number of vaccines per citizen, or number of doses per citizen, with the number of companies that they've signed. So one day someone's going to have to pay for all this, but nonetheless, there are a lot of vaccines available in Canada and a lot of pre-purchase vaccine. And hopefully we'll be able to see all of the figures brought forward by the Canadian government at some point in the future. With respect to the applications to date, we know that we have Moderna and Pfizer Canada that are, and sorry, this slide is a date I should have put, that is authorized the Moderna vaccine, and then the Pfizer Canada, the BioNTech vaccine. Janssen and AstraZeneca are under review. I expect that the AstraZeneca, which is actually a respiratory virus vaccine vector type of vaccine that is going to be made available. That's the one in the UK, the AstraZeneca Oxford University vaccine, which will be coming available. And I think it'll be next in line to be approved. And with respect to making these vaccines, the messenger RNA, this is just a little cartoon, and I like to describe this as an M&M candy. So, because many of you have heard there's this cold chain, it's got to be kept at -70 plus degrees, -70 to -80 for the Pfizer vaccine. And then the Moderna at -20. Pfizer believes that they will get to -20 at some point in the future. But what it is, and I use the M&M, and pardon me for trying to take an analogy on this, but just to explain what's happened with the messenger RNA is it delivers the messenger RNA, which is used to be able to turn the body's cells in the muscle where it's injected into a manufacturing piece to be able to build the antibodies to the messenger RNA, the message that is brought in for the coding of the genetic code, bringing it to the building blocks, which are the protein manufacturing factories, if you like, in the muscle to be able to produce the antibody. But it's a very sensitive piece of machinery when you think about messenger RNA. There's these long strands, if you remember your basic DNA and how it comes with the cross linkages. It's very fragile, and so what happens is that they coat it, and it's like an M&M candy. So the chocolate, the piece that you want is on the inside, but they put this outer shell on it, and then it's got to be kept cold, because if the temperatures, it's very sensitive to freeze thaw, and if it ever was left out too long, so many of them vaccines with Pfizer and Moderna, after they're taken out of the freezer, you have to allow them to thaw over several hours, and there's timing. Then they have to be reconstituted, then they have to be used within the Pfizer vaccine, for example, 6 hours. Otherwise, it starts to be rendered ineffective. If you don't use it, it has to go back in the fridge, and then after another 12 hours, it becomes inactive, and that's where you hear about, oh, they're wasting vaccine, and why they're so insistent on having set appointments for people to come in and not to miss their appointments, or to do what they're doing in Israel, where now 25% of the Israeli population has been vaccinated. And that's because they've initiated an almost military-like precision with 24/7 vaccination centers. They work all day, all night, 7 days a week, and they're just bringing people through to vaccinate. And so that's the issue with these messenger RNA vaccines, is that to maintain the stability so that you can get the 95% protection, you have to keep this cold chain, and that's because they have coated the virus with this different types of fatty coatings. And these fatty coatings, and I'm going to touch on that in a moment, are the ones that protect the inner piece, so like the chocolate in the M&M, and then you've got this hard candy coating around it. And that's why it's important in the delivery process with these vaccines, and so to help you understand about this cold chain issue. So it's a major limitation, but not one that cannot be overcome, because if you think about Ebola, and they were using this in the tropics, they were able to maintain cold storage. So surely in a country like Canada, we should be able to do it. And I think they're doing it, but it's just presented some challenges in the beginning. So with the first vaccine, and I won't take you through all of this, but this is a classic phase 3 study where they did the study design, carefully chose the population, the outcomes, looked at the efficacy, the safety. It's a very rigorous trial to do a phase 3 trial, and then looking at the storage and how well it was stored, and how it can be delivered. So there were 2 doses given 21 days apart. And you've probably heard on the news that the WHO and other organizations, and even Pfizer and BioNTech, the German company with whom they partnered, have indicated that you can have an initial response, but that the second vaccine dose can be delayed up to 42 days to allow you to be able to have a satisfactory response. And this is the bottom line, and I'll just highlight it here for you, is that basically you're looking at 95% effectiveness in this vaccine, consistent across age, gender, race, ethnicity, and even in the elderly over 65, it was 94%. So very important. There are trials now underway as we speak in children and in that are less than age 12 and in pregnant women. The Obstetrics and Gynecology Association has come out with a statement that it is a safe vaccine. It's a non-live active vaccine, so it would be safe in pregnancy. So this is really a great finding that we have a vaccine that appears to be so effective in this regard. I mean, you think about influenza, it's about 50 to 70% effective. And as you get older and older or have multiple medical conditions, it decreases the impact of that influenza vaccine. The Moderna is very similar. It's a messenger RNA using a non-replicating RNA. And it's, again, entrapped in this sort of fatty outer layer, much like I use the analogy of the M&M candy. And then the chocolate or the internal part has got the messenger RNA. And it also has a cold chain, but at -20. Moderna has a little bit of an edge on Pfizer in their technology because they've been working at this for a longer period of time. And their outcomes were very similar, 95% effective in a very large phase 3 trial. And again, pretty good tolerance. I didn't touch on it with Pfizer, but they're very similar. There were a number of people who had side effects after the second dose, fatigue, aching, a sore arm, redness at the injection site. Remember, this is built to be able to induce a fairly stringent and robust antibody response. That's why it works so well. So that's sort of a signal that it's going, and it's, I liken it to when you had your first influenza vaccines, you know how you have a sore arm and you might get a bit of aching for a day. The more serious effects, and I'll touch on that in a moment, are those that have allergies. But in general, it's an extremely well-tolerated vaccine. The AstraZeneca vaccine is a chimpanzee adenoviral vector vaccine. So they took a part of the chimpanzee adenovirus and they're using that as a vector to be able to provide the immune response, following the injection. A couple of things on this is that with their study population, they did look at children ages 5 to 12, and they looked at similar items in their outcomes. But in their study, there was an error in dosing almost 3,000 participants in the UK, and that they were using half of the dose in terms of the pooled data. It had erroneously given half dose, but yet it had 90% efficacy. And then when they gave the planned 2 doses in almost 9,000, there were a lesser effect with only a 70% vaccine efficacy. They're still trying to sort that one out. People say, well, it was a fluke that they gave only half the dose for the first dose. So, it showed that it was more highly effective than maybe they were overstepping it with the amount that they were using in the first dose. And this trial was halted for a period of time because they had a side effect with spinal paralysis in one of the patients. And it raised some concerns about a swine flu type, if you remember the paralysis issue that occurred with the swine flu vaccine a few years ago. But there's been no cases since then, so they think it was probably a one-off fluke. And with the National Advisory Committee on Immunization Guidance, the key populations, and I'll touch on this in a moment, are the high-risk populations, meaning those over 65 and those with advanced high-risk conditions. Still evolving, but we know what many of them are. And then secondly, those most likely to transmit coronavirus 19, which are healthcare workers, caregivers, household contacts. Then next are police, firefighters, grocery store workers, and then indigenous communities, those in crowded conditions and correctional facilities. The risk factors, and I'll just touch on this because this comes into some issues about lymphedema. So age is probably the major one, those 65 and over. And I think you probably know that. And then obesity with the body mass index over 35, diabetes, and they both have higher rates of hospitalization and also ICU admission and higher death rates. Pregnancy is also associated with a higher risk of intensive care admission, probably as a reflection that in pregnancy you have a relative degree of suppression of the immune system because women are carrying that baby. And then some of the underlying conditions, chronic heart failure, chronic lung disease, and then the prevalence of these multiple comorbidities, so diabetes, advanced kidney disease, pregnancy, obesity, they are associated with worse outcomes, and then certain types of cancers. And this is something that's important for those that may have lymphedema post lymph node dissection or following breast cancer types of surgery. And in lymphedema specifically, lymphedema alone, and I've searched through the literature and all of the other settings, it alone does not make you more susceptible to the coronavirus and illnesses. So that's a piece of good news. There is one exception, and this is individuals that may have lymphatic abnormalities involving the chest and lungs. So we do know that there are some people that have had lung surgery and they've had to tie off the main lymphatic ducts that carry the lymph channels. So there are certain conditions, thankfully it's not very common. And then if you have lymphedema associated with some generalized immune deficiency, so if you have some deficits in the ability to produce antibodies for whatever reason, and lymphedema, that would put you at risk, but it's not due to the lymphedema itself, but more related to the B cell deficiency in the ability to be able to make those antibodies. So that would be a condition such as myeloma, or there's rare conditions where you just have lowered levels of antibodies in your body. I would say the major issue with lymphedema, and some of you may have experienced this if you are individuals with lymphedema, is navigating the health system in the COVID era. It's been so difficult, not only for individuals with many types of medical conditions, but with lymphedema, and then also the issue of the hygiene of high-touch surfaces because of the procedures that are used with the massage therapy and then the lymphedema stockings. One needs to take great due care and attention with respect to ensuring that these are cleaned properly in detergent. The virus is enveloped, and thankfully it's prone to any heat over 40 degrees centigrade and also 40% alcohol and above. But we do know recent studies showing that it is present in the environment in a major way. We've done some of those studies here in Alberta, linking in with the level 3 laboratory at the Li Ka-shing Virology Institute in Edmonton. One is now just opened up through the Snyder Institute in Calgary, but they've been doing cultivatable virus studies since March in Edmonton, and we've sent them over 60 patients and have shown, and we've not published this yet, but we've shown quite significant presence of cultivatable virus, including on persons' hands, on objects, cell phones is one, and in other samples. So we know this virus is present in the environment. And then finally in Alberta, and I'm just going to close, there is a task force led by Deputy Minister Paul Winnick for many months now in terms of the planning, and they have a multi-phased approach for delivery beginning in December and following through into September and the fall, where I think they're hoping that the population will be immunized in a general sense. So that would be anyone with lymphedema in the spring, late spring to summer. I know that's going to affect your conference that's being planned and other things. So I think by 2022 we'll be in good shape. The vaccine may have to be given. They're talking about once a year now. They're looking at it maybe every 2 years. So that gives us a window of opportunity. And as I mentioned, in Alberta, they were hoping to provide almost 30,000 doses by the end of December. That was not met. They were expecting a big delivery of Moderna vaccine in December. It did come, but it was in that holiday period. But The good news now is that as of January the 12th, so that's yesterday, they have done some catch up and they've administered 53,100 doses. And in fact, today, you've probably heard, have started canceling appointments because they're running low on vaccine. So they've really ramped up the vaccination, and it is impressive. There are about 80,000 to 90,000 healthcare workers in AHS, and almost 25% have been vaccinated. Long-term care and designated living staff, designated support living staff, they've done almost 18,000 and 16,000 long-term care residents. So moving along reasonably well. First Nations is another targeted area, as are EMS. And unanswered questions, just very quickly, children, pregnant women, but the Society of Obstetricians and Gynecologists have said in their statement that's just been recently released, they believe that pregnant women should be allowed. It's a safe vaccine in pregnancy. And I think most of us believe that that would be the case. Children remains a question, but I expect as they finish with the adults, children, for many reasons, appear to have less severe illness with the COVID-19 than adults. And that's still not understood. Perhaps the Snyder Institute and their immunology group will be able to provide those answers. Safety concerns, less than 1 in 10,000 for major issues. There are no specific safety concerns in lymphedema. You'd be happy to know, even with the injections. And there are trials going on. I just want to touch on this for a moment, with multiple companies recruiting in children from ages 6 months forward. So we will hope to see those answers in the ensuing months. And with respect to the major side effects in the United States over the weekend, they've now recorded enough allergies. They've come out with a rate that there is an allergic reaction in 11 in 1 million. So that's pretty rare. That's about 1 in 100,000. And it has always been in individuals who have got significant allergies. Why is that? So if you've got shellfish or peanut allergies, or you've got multiple allergies to medications and or other environmental items, remember that there are 4 fatty substances that coat remember the M&M analogy, that coat the outer part of the messenger RNA that carries this. And there are several salts that are present, and they also use polysorbate, which is a derivative of sorbitol, and sorbitol is in a lot of as a sweetener. And so there are some breakdown products of sorbitol. So there are some people that are just more sensitive to certain chemicals and or items, and I think that's why we're seeing the allergies. Most of the individuals have responded to antihistamines and or their EpiPen injections. So no one has had a serious intensive care hospitalization to date, and that's with millions of these doses given. But that's the story. You might read in the media, oh my God, someone almost died from the vaccine. Well, that's not the case. If somebody forgot their EpiPen and decided to get a dose, hundreds of miles from the nearest medical station, and there was no EpiPen, there might be serious consequences, but it would be no more than if you had food stuff that somebody inadvertently dropped peanut dust and you're highly allergic to peanuts into your food. So that's why it's recommended that you carry your EpiPen if you have allergies. And in conclusion, astonishing global progress in the development of these vaccines. It's been rapid, I would say not rushed, and the science has met the mark. There are unanswered questions, pediatrics being one of them, and then a secondary question is this first versus second dose timing. And then a third question, if you're immunized, how soon after you've had COVID can you be immunized? That answer is coming forward. In the last 24 hours, I've learned probably 2 weeks after COVID recovery, you can be vaccinated if you're eligible. And then finally, if you become infected with COVID again, how soon do the antibodies ramp up in your system? And we do have some data from the non-human primate studies with the macaque studies, if you know that variety of the monkey population. And it shows in those that have not had vaccine but have had natural infection and then have been re-challenged with live virus. The live virus can hang out for a few hours and then it's immediately attacked and knocked out of the body's system. And that's the closest that we have to human data. People are suggesting that there's at least a 70% chance that the virus would be knocked out in hours. But that has an important implication, which I'll show in the last slide. Even in the era of getting vaccinated, you need to still use those precautions, which is the distancing, which is probably one of the most important items. You need to use your frequent hand hygiene, your hygiene of high-touch surfaces, and your masking, and a good dose of common sense with respect to gatherings and other things. So it's not like you can be vaccinated and then I'm going out on a shopping trip and I'm heading to Hawaii and I'm going down into the thick of things in the United States because I'm in a protective cocoon being vaccinated. I think one needs to exercise some common sense and allow the vaccine to play itself out so that we develop this herd immunity, not only within Canada, but globally. And with that, I'll close. Thank you for your attention this evening.
Wonderful. Thanks so much, Dr. Conley. That was a really, really interesting presentation. Really appreciate all the insight and the data and knowledge that you've shared. I do have a couple questions here from our audience members. So I'll read the first one out here. How long does COVID stay on, say, fabrics, something like a compression garment? Any suggestions for patients who have to touch surfaces while wearing a compression glove as a result of arm lymphedema?
That's a great question, and I touched on that because I was predicting it would come up, and so I was correct. So we've done some studies in hospital. This is unpublished material, and we have actually looked at in the Li Ka-shing Virology Institute with our colleagues in Edmonton, you know, the fabric masks and then also the N95, and they've dropped virus at about 10,000, they measured in plaque forming units per milliliter. And then they've come back several hours later, and the virus is merrily sitting along its way. We also did a study on a patient in hospital who had a recent organ transplant, and so they were their immune system was quite depressed, and they carried cultivatable virus for 33 days. And we took and we placed it onto a series of Petri dishes. So I'll bring in my Petri dish here. And we dropped spittle about the size of a thumb. And we had to just drop the spittle. We let it dry out and came back some hours later and reconstituted. I did this experiment in a patient's room because we can't do human challenge experiments. And we found 10 to the 4th plaque forming units per little thumbnail of a spittle in both a wet sample that we kept the viral media in, which is optimal growing conditions. And then and it looked just like a child, you know, if they spit on the ground and you say, oh, why are you doing it? Or a dried piece of gum. And we thought nothing of it, but it was loaded with virus. So, and that was several hours after. So we know from other studies that have been done on surfaces, and this virus tends to love plastic. With cloth, it often soaks in and then it will soak into the cloth. And they have been able to find virus on the internal surfaces of cloths. So I think we're probably measuring it in several hours, but certainly if you have, and I can tell you that in our studies, saliva and nasal secretions contain large quantities of virus. So if you've got somebody who's sneezing, coughing, spitting, those droplets contain large amounts of virus. And if it gets on surfaces, including any of the you know, stockings or sleeves that are used with lymphedema, you need to immediately remove them. If you had somebody who sneezed or coughed on it, do hand hygiene and then take it and put it through a hot water cycle at 40 degrees or higher in that respect. So it's always a good idea to carry an extra sleeve in case you're going to be out and about or there are other things, and then you're prepared in the event that occurs. So I think it's just prudent planning, much like a healthcare worker who carries an extra mask with them. We all are doing that nowadays. So that's important to recognize, and that if you think you've been exposed, and again, those secretions, saliva and nasal secretions and any phlegm. I'll give you one other example. In one elderly patient, I'm still an active clinician as an infectious disease physician. I've seen over 100 COVID patients myself personally, so I'm very experienced with COVID as an illness. She coughed up a big, thick piece of phlegm, you know, that came down from deep when you've got something rattling in your chest, and it flew out and was laying on the gown. And a few minutes later, the nurse knew I was collecting specimens, and at the door said, "This patient just coughed up something. I think it might be a specimen you're interested in." And it was very thick and gooey, and I had to scrape it off with a tongue blade. That grew 1 million plaque forming units per milliliter. And so that was lying on a patient's gown. So this is not published. I'm giving you very high level unpublished information. I'm just trying to put it together, but we have been doing this. I know some of the literature has suggested that, oh, the environment's really not that important. Why are we doing all this touching the elevator button and cleaning it? Well, our data is going to change the scope on this. It is important, no question about it. Just like it transmits like the common cold in many respects. And in fact, it's a kissing cousin to many of the human coronaviruses, which cause about 30 to 35% of all human colds. So a long-winded answer, but important.
Absolutely. Thank you for that. That was very interesting. A couple questions here about kind of site of vaccination with lymphedema. You know, it's a common topic, and we got a couple questions about the first one here is, if a person has, say, lymphedema in both arms, can they still receive the vaccine in another site, such as I think they put the
yes,
thigh here.
That is similar to any other site where you might be using. There are individuals that, for whatever reason, have a problem with an arm, or if they're a dialysis patient and have a fistula in place and they have quite a swollen arm. Remember, it's given in an intramuscular, so they can actually get through into the muscle. And to my knowledge, and I was prepared for this question as well, because I thought some clever person's going to ask it. The lymphedema is more in the vessels, and so within the muscle, with the mechanism by which when it's in the muscle itself, even if you had some swelling in some areas of lymphatic fluid that was present, when it gets into the muscle cells, it turns those cells into its manufacturing factory to be able to produce. So it should not be impaired by any leaking out of lymphatic fluid. So you probably could have it in the arm, but if you had another site, just as any vaccine, you could receive it at any site. You could get it in one of your cheeks and in the back end and the buttocks. I have seen patients with congenital lymphedema of the buttocks, so that's a rare condition. But I had a patient who had recurrent cellulitis that I was seeing in the clinic, and it was a difficult one to manage because they always had one side of the buttock more swollen than the other, unfortunately. So that would be an area to avoid as well, because you always have the concern, is the needle long enough, and if you have a swollen arm. So, but generally they are able to get into the muscle. So, but great question. Thank you for that.
Absolutely. So yeah, I think we have time for maybe a couple more questions here. One came in stating, if I received the 2 doses of the vaccine, how safe is it to travel, and how likely is it that I could be infected again?
You know, pretty unlikely you'll be infected, 95% protection. Even in those over age 65, it was 94%. So the likelihood that you would get infected is low. It's not impossible, given that, you know, it wasn't a 100% effective vaccine, but once the large majority of the world's population, or let's say in Canada, are vaccinated, you have this thing called herd immunity, which once you get, and people throw about different numbers. Dr. Fauci says 90%, our medical officer of health says 70, but somewhere in that range of 75+%, the virus can't continue to propagate and it sort of dies out in the population. That will be a signal that there's a great safety to travel. I think we're all looking to get out of our cocoons, but there may be a little bit of a wait. It would be considered relatively safe, you know, in that respect, particularly if you had a good antibody response, which the majority of the population would. I don't think it gives you an immunity passport and you book your trip to a sunny destination as soon as you're vaccinated and get your test done. Some people will choose to do that. I probably will wait a little bit and wait till a large segment of the population is immunized, but you should be relatively protected in that respect. I would say it would still be prudent. You don't want to go into a large-scale gathering with Trump-like people and decide that you want to test yourself, how good is this vaccine? I don't think I would march into that type of a setting, and just use some common sense. But it probably will be safe, bottom line.
Fantastic. And we'll just do one more question here. So we talked about kind of susceptibility in terms of lymphedema and COVID, but there's also a question here from someone who they've talked about how they've noticed that their lymphedema can worsen after they have, experience a virus themselves. And they were wondering if there is any evidence or any risk of lymphedema actually worsening after someone were to contract COVID.
Okay, that's one I hadn't thought of, and I didn't even see that in the list of in the product monograph, because in those 10,000 people, did someone have lymphedema? So that's a great question, and it's something that you might want to take on in your lymphedema association and begin a registry, perhaps, to say for those of us who are being vaccinated, how many experienced an increase in their lymphedema state after vaccination, or even looking after a viral infection. We know that there can be after effects from any severe viral infection. We've all had influenza or severe colds, and we feel a little bit, sometimes you get swelling in your legs, even normal people without lymphedema for whatever reason. So, but it might be something to try to look at and even have some type of questionnaire or mail-in ballot of some sort from your lymphedema association following the vaccination. It might be worthwhile looking at that, and perhaps the Alberta Lymphedema Association can provide that answer for the others across the country.
For sure. I know we have a couple of folks from the Lymphedema Research and Education Network too. So there's a couple of researchers from the ground. Yeah, absolutely. All right. Well, with that, we're just nearing 8 o'clock, so I think we'll close off our event tonight. Thank you all for joining us for tonight's Lymphedema Night, and thank you so much again, Dr. Conley, for sharing your knowledge and insight.
Oh, you're welcome. I hope you found it useful. I'm sorry I couldn't do more lymphedema specific, perhaps at another occasion. We'll talk about lymphedema and infection, which is loaded with lymphedema issues, but I thought this was a topical area, and I tried to touch on some elements, at least to reassure individuals that lymphedema is not anything that should be any concern with respect to COVID-19 or COVID-19 vaccination.
Absolutely. Yeah, and I think it would be great to have you back for that situation. Thank you so much. Yeah, and so with everyone, we just want to invite each of you to check out our association on Facebook or our website. We will be announcing some upcoming Lymphedema Night events in the coming, hopefully in the coming months, for us to take part in. I know we look forward to connecting through this series again soon with all of you. So I will end off our event here by just saying take good care, be safe, and be well, everyone. Thanks very much for your time, and have a great night.